Hello guys, now let's begin with the new chapter, that is genetics. So first let's begin with what exactly is a gene. See, you know that in every cell there is something called as a nucleus. Inside the nucleus what do we have? We have the chromosomes. We humans, we have 46 chromosomes, that's 23 pairs of chromosomes, right? Now let's take a chromosome. Now, if you just decondense it, if you just open the chromosome, what do you have? You are going to have the chromatin, and at the end of the day, what you are going to have is this DNA. This DNA, it is compressed into the chromatin, and the chromatin is going to form the chromosome, right? So, in this DNA, what do you have? A segment of a DNA, okay? A segment of a DNA or a series of nucleotides is called as a gene, okay? So what exactly is a gene? It's a sequence of certain nucleotides. First let's begin with, so what exactly is a gene? It is sequence of nucleotides. It's a sequence of nucleotides which is going to be result in some protein formation or some certain character, okay? So it's a gene. So how many genes do we have? We almost have 20,000 genes. We humans, we have the 20,000 genes, okay? Now, what exactly is this term exon and introns? Exon means the expressed genes, those genes which are expressed, okay? So, those genes which are expressed are called as exons, and those genes which are not expressed, okay, not expressed are called as introns. Okay, those genes which are not expressed are called as the introns. So exons are genes which are expressed, introns are the genes which are not expressed. Okay, so we have lots and lots of genes which are not expressed, they are just silent, they are just like that. Okay, they are not expressed. Maybe they might be working in the evolutionary process. Okay, maybe 10,000 years back, 20,000 years back, 30,000 years back, they might be working, but right now they are not working, they are simply silent. So they are called as the introns which are not expressed. Now after this, the other term which I want you to know is called as the locus. Okay, what exactly is this locus, sir? Locus means, for example, there is one gene, I am talking about one gene, X gene. Where is it located? On which chromosome number it is located? On which arm it is located? Whether it is located on P arm or Q arm? If it is located on P arm, where exactly? Which band? Which subband? So the address of a gene is called as locus, or I can say it's a location of a gene. Location of gene is called as locus. For example, let me show you here. See, there is a gene called a CFTR gene. Okay, cystic fibrosis transmembrane regulator gene, CFTR gene. See, the location of this gene is 7q31.2. 7q31.2. What does it mean by? It mean by, see, this CFTR gene, let me put it here, CFTR, it is present on chromosome number 7. The first letter it indicates chromosome number 7. Okay, it is present on CFTR gene is present on chromosome number 7. 7q, what is that Q stands for? Q stands for which arm? 7 stands for which chromosome? Q stands for which arm it is located. And 3, what is this 3 stands for? See, this 3, it is standing for which band? Okay, let me show you here. Guys, look here, there is band here, this is a band and this is subband. Okay, band, subband and region. Okay, so band, subband and region, there are 3 things which I want you to know. Here, what is this 3 standing for? The 3 stands for first which region. Okay, 3 stands for which region? So in this region. Okay, in this region. So let me write here 3 stands for region. Okay, 31. What is this 1 stands for? 1 stands for band, region band, and 31.2. What is this 2 stands for? 2 stands for subband. Okay, so what I want you to know is, so what exactly is the locus? Locus is the address of a gene, okay, or the location of a gene. So if I say CFTR gene is present on chromosome number 31.q, 7q, okay, 7q31.2, which means chromosome number 7, Q arm, region 3, band 1, subband 2. Okay. For example, there is one more gene which I will give you, which is very much important. There is a syndrome called as DiGeorge syndrome. So, DiGeorge syndrome. If I say in DiGeorge syndrome, 22, the genes which are present on 22q, 11.2 genes, these genes are deleted. Which means what you can say now, so there is a chromosome number 22. On this chromosome number 22, there is a Q arm, there is region number 1, band number 1 and subband number 2. So, these genes are deleted, which is leading to the DiGeorge syndrome. So, locus is the location of a gene. Okay. So, after this, the next point which I want you to know is allele. What exactly is allele? Here itself I have already written, allele means alternate forms of a gene. Okay, alternate forms of a gene. Now, let's take one, for example, let's take one gene, and that gene is responsible for my height. Okay, there is a gene which is responsible for my height. So, height gene or my color gene, height gene, color gene, or gene for enzyme, or gene for the color of my eyes. Okay, anything you can take a gene. So, this gene for height have two phases or many phases, you can say. There are many phases, but mainly we are talking about the two. The alternate forms. See, a person, if I ask what is the person's height, he may be tall or he may be short. The two alternate forms of a gene are called as allele. So, one is short and one is tall. So, short and tall are the alleles for height gene. For example, gene is, for example, color of my eyes, there is a gene for the color of my eyes. It can code for black color, it can code for green color, blue color, yellow color, some other different colors. So, the possibilities, the alternatives of a gene are called as alleles. For example, look here, see there is a gene, imagine there is a gene, it's coding for the eye color, it can code for blue color or it can code for brown color. So, there is a gene for, see there is a gene for hair color. So, what are these? This yellow, red, brown, black, all these are the alleles, the alternate forms of a gene. It can be many. So, some books will say there are only two alternate forms, but many alternate forms can be there. Okay. So, here there is a gene for eye color. See, there are brown eyes, blue eyes, green color eyes, grey eyes. Okay. They are all the alleles. See, alternate alleles. These are the gene, these are the alternate forms. So, now you know what exactly is the allele. Allele means the alternate forms of a gene is called as allele. Okay. Now, after this, let's discuss about the terms like homozygous and heterozygous. See, this is an introduction video, so you should know all this terminology. Okay. So, what exactly is a homozygous and what exactly is heterozygous? See, we humans, okay, we have how many chromosomes? 46 chromosomes. So, out of that 46 chromosomes which we have, how many are from mother? 23 are from mother, 23 are from father. Okay, 23 genes from mother, 23 genes from the father. Okay, you know this, right? So, we have 23 mother chromosomes, 23 father chromosomes. The point which I want you to know is, on chromosome number 1, for example, if I take chromosome number 1, how many chromosome number 1s will be there? 2 chromosome number 1s will be there. 1 chromosome is coming from the mother, 1 chromosome is coming from the father. This is chromosome number 1 from mother, chromosome number 1 from father. So, 1 chromosomes will be there, 2 chromosomes, 3 chromosomes will be there. Something like that. Okay, 23 pairs are there. Now, the point I want you to know is, look, on the chromosome number 1, for example, I am telling you, here is a gene for height. The same locus from the father here on this chromosome also, same gene for height is there. So, this gene is coding for the height, this gene is also coding for the height. For example, look, now here mother is coding for short. Now, mother is short and she is having an allele. See, now she is having a gene which is coding for short height. Now, I am just representing like small t. Father is tall, he is giving an allele capital T. Okay. So, what are these two? These are the two alternate forms of genes, the two alternate forms of the genes. Okay. Small t, capital T. Okay. These are the two alleles for the height. Now, see, in the baby, okay, in the see, mother is giving small t, father is giving capital T. So the person is going to have something like this, capital T, small t. So this is called as heterozygous state. Now the person is heterozygous for the height genes. One capital T is there, one small t is there. So this state is called as heterozygous state. So let me write here, heterozygous means he is having two different alleles, one capital T, one small t. He is heterozygous for the gene. Now homozygous means, for example, if he is having capital T or small t, if you have same alleles, one from father, one from mother, one from father, one from mother. If you have same type of alleles, then it is called as homozygous. If you have different alleles, it is called as heterozygous. Okay, so homozygous, heterozygous, now you understand. Okay, now after this, let's discuss about what is the dominant allele, what is the recessive allele. What exactly is the dominant allele and what exactly is the recessive allele. Okay, so dominant allele means which is expressed. Okay, see, always there are two alleles. Okay, in a person there are two alleles are present. Okay, which allele is expressed, that is called as dominant. For example, let me take a person. See, this is the person. Now this person is here like this. Now let's see. So this person is having capital T and small t. For height gene, he is having two alleles, capital T and small t. Mother is giving small t and father is giving capital T. Now he is definitely heterozygous. In this heterozygous state, whichever allele is expressed, that is called as a dominant allele. Now this capital T allele is, this person definitely is going to be tall in height. This person definitely is going to be tall in height because this allele is dominant, capital T allele is dominant over the small t. So in the presence of heterozygous state, whatever the allele is expressed is called as dominant allele. This small t, it is not going to be expressed. So this is called as recessive allele. So dominant allele hai and recessive allele hai. So now what exactly is dominant allele, I have explained you. So whoever is expressed, it is called as dominant allele. A recessive allele means do not express, it usually do not express in heterozygous state. In heterozygous state it is not going to be expressed. Then when it is expressed, sir, only in homozygous state it is expressed. Okay. Now for example, see again there is this one person. Now from father and mother, okay, from father and mother he is getting the small alleles only, small t, father is short, mother is short, both of them they are giving small t. Now this person, now these alleles are going to be expressed, he is going to be short. So this allele is only expressed in homozygous state. But dominant alleles are expressed in heterozygous state, as simple as that. So recessive alleles expressed in homozygous states. For your understanding purpose, let's take an example. Okay, let's take an example. So here mother, father, let's take mother and father. Okay, mother and father. See, mother is having these kind of alleles, capital B, small b. Mother is heterozygous. She is having capital B, small b. Father is also heterozygous, capital B, small b. Whatever I am showing you here in the capital, these are the dominant alleles. Capital B means dominant allele, small b means recessive allele. Okay, capital B codes for brown eyes, small b codes for blue eyes. You see, capital B, it's a dominant brown allele. Small b, it's a recessive blue allele, blue color eyes. Now, this couple, if they have kids, what kind of combinations can be possible? See, there is a possibility that in one kid, he can get capital B. Means both the dominant alleles. If both are dominant alleles, definitely brown eyes. Okay, definitely brown eyes. In one kid, the person can get one capital B, one small b. So one capital B, one small b. Now look, one capital is there, one small is there, one dominant is there, one recessive is there. Who is going to be expressed? Definitely in heterozygous state, who is expressed? In heterozygous states, sir, definitely dominant allele is going to be expressed. Okay, so look, dominant allele is expressed, brown eyes are there. Okay, even though the small b is there, but still it is recessive. Its actions are suppressed by the dominant allele, brown eyes. Now, same thing, capital B, small b. See, it's a heterozygous state. Capital B is there, so definitely brown eyes. But recessive alleles are only expressed in homozygous state. If there is a recessive homozygous state, one small b, then only they are expressed, blue eyes can be possible. Okay. So, recessive alleles do not express usually, they are expressed only in homozygous state, small b, then expressed, small t, then expressed. Okay. So, this is the point which I want you to know. Next, sir, there are certain genes which will be having codominance, which will show a property called as a codominance, sir. Sir, what exactly is this property of codominance? Kya hai? What exactly is this? For example, let's take this example, weird example, but this is in most of the families. See, in a family there is father and mother. Okay. Usually, one person will be dominant. Okay, father will be most of the time dominant. Okay, most of the time his thoughts, okay, are going to be expressed. Okay, but there are certain families where both are dominant. It's not like there is no such partiality. Both mother and father, they are like taking their own decisions, and like whatever father says and whatever mother says, at the end of the day, both will happen. Both will happen. Okay, there is no dominance, particular dominance. Both are dominant. So there is something called as codominance. Means I have said you, usually one of the allele is expressed, but here in codominance, both the alleles will be expressed. Okay, both the alleles will be expressed. See, both the dominant alleles are expressed. For example, here both the alleles are dominant. That's what I mean by. Okay. For example, this is seen with ABO blood grouping, and HLA genes. ABO blood grouping and HLA genes, they are example of codominance. So what exactly is this ABO blood grouping, sir? See, these are the different alleles A, B, O. These are the alleles for the blood grouping. There are three alleles A, B, O. Father is also having ABO, mother is also having ABO. So these are different alternate alleles. Now, if they have kids, for example, see if they have kids, this mother and father, they have three different alleles for this blood grouping. Now, if they have kids, what are the possibilities? See, one kid can have AA, something like this AA, so definitely the blood group is going to be A. One kid, see, I have said you, these alleles, this A allele as well as B allele, A and B, both are dominant, only O is recessive. Okay, only O is recessive, O allele is recessive. A and B are dominant. If a baby is getting A from mother and B from father, both are going to be expressed. So the blood group is not going to be A, the blood group is not going to be B, the blood group is going to be AB blood group. Okay, understood? See, for example, a baby is getting O from the father, A from the mother. For example, A is coming from the mother, O is coming from the father. A is dominant, so A is expressed. Okay, only A and B, A, B are dominant alleles. Both A is dominant as well as B is dominant, but O is recessive. In the presence of A and B, for example, a baby is having both A is there, B is there. Both will be expressed. So it is AB blood group. Okay, so it is AB blood group. See, mother is giving B, father is giving A, so it is AB. In the presence, see, for example, B and B both are coming, so it will be B. Both are dominant, B, dominant. So mother is giving B, father is giving O. B is dominant over the O, so B blood group. So what I am trying to put into your mind is codominance. The property of codominance is seen with ABO blood grouping. ABO blood grouping is an example of codominance. Both A and B both are dominant. In certain families, both father as well as mother are dominant. So equal thoughts. Okay, both are going to take the decision. Codominance. Okay. So next, after this, the next point which I want you to know is genotyping. Okay, what exactly is the word genotype means? Genotype is a genetic makeup. A genetic makeup of organism. Okay, it's a genetic makeup of an organism. So what kind of genes are there, what kind of alleles are there? It's a genetic, totally genetic thing. So it's a genetic makeup of an organism is called as genotype. For example, me. Am I having capital T small t, or one capital T and one small t? So what is the genetic makeup which is present within the cells? You cannot see it directly. That is called as a genotype. Then what is phenotype, sir? My appearance, what you can see, what is getting expressed. So phenotype is what you can see. Genotype is what is the genetic makeup. Okay, what is the composition? What is the genetic composition is the genotype. what is getting expressed is phenotype. For example, look, just look here, genotype versus phenotype. See, genotype of an organism is defined as actual or complete genetic makeup of an organism, whether it is capital T, small t, or small t, or whether capital T, what is there within the genes is called as a genotype. Okay, genetic level. What is a phenotype? Phenotype is what is getting expressed, whether the eyes are blue or black or yellow or green, whether the person is tall or short, the physical appearance. See, phenotype refers to individual observable trait, what you can see directly. Observable traits such as height, eye color, blood type. A person's phenotype is determined by both their genetic makeup as well as environmental factors. That's the different thing. Okay, phenotype is dependent on the genotype. It's the genotype which determines the phenotype as well as the environmental factors. Okay, according to the environmental factors, the genes are going to work and determines the phenotype. Okay, here also say, for example, this is the genotype. For example, this is what you study in your botany in the class 11th or 12th. This is the genetic makeup. Okay, capital B, small b, capital B, small b, small b. It's a genotype. Based on the genotype, you can have the different colors. Phenotype, this colors expressed, what is getting expressed, the color of the flowers. It's called as the phenotype. Okay, so genotype, phenotype is completed. Now, after this, now let's discuss about the mutations. The basic video, you should also know about the mutations. What exactly is a mutation? Mutation means permanent change, a sudden permanent change in the DNA material. It's a sudden permanent change. So, sudden permanent change in DNA material. Sudden permanent change in the DNA material is called as a mutation. Now, in this video, what you should know, how many types of mutations are there? See, mutation means what? There is some change in the DNA material. DNA material got little changed. So, that's going to show, definitely that's going to cause effect, right? So, how many types of mutations are there? The first mutation for your exams is the point mutation. Very, very important. Point mutation, where is a small mutation? Point, at small level, there is a mutation. Point mutation is there. So, how many types of point mutations are there? Okay, there is one single change, one nucleotide change. Okay. So, what exactly, how many types of point mutations are there? Actually, there are 3 types of point mutations. What are the 3 types of point mutations? One is silent mutation, nonsense mutation. Okay, nonsense mutation, and third type is missense mutation. So, there are 3 types of point mutations. One is silent mutation, nonsense mutation, as well as missense mutations. So, what exactly is a silent mutation? Silent mutation, what exactly is it? See, for example, you see, here I am showing you point mutation. This is the normal original sequence in the DNA. See, this is a DNA strand. Okay, this is a DNA strand, one strand of the DNA. Now, in this DNA strand, see, all these are the nucleotides, right? They are the different nucleotides. For example, imagine this is a gene, a sequence of nucleotides. This is a gene, sir. Okay, this is a gene. So, gene for something, gene for eye color, or gene for some enzyme or protein or something. This is a gene. Okay. Now, in this gene, see at this place, okay, at this place, for example, this area, it was supposed to be T. There was supposed to be thymine. Okay, there was supposed to be thymine. But instead of thymine, there is C. Okay. So, instead of thymine, you have adenine, guanine, thymine, cytosine. These are the nucleotides, right? Instead of thymine, there is now here cytosine. Cytosine is there. So, it's a point mutation. One point was changed. So, how many types of point mutation I have explained to you? There are 3 types of point mutation: silent mutation, nonsense mutation, and missense mutation. What are the differences between these 3? Look, first I am going to explain you about the silent mutation. Sir, what exactly is silent mutation? See, in the name itself it's there. See, though mutation have happened, though mutation have occurred, but it is absolutely silent. There is no problem, absolute silence, no problem. See, it was supposed to be GAA, the codon was supposed to be GAA. So, it was supposed to be, the codon was supposed to be GAA. So, what happened? Instead of this A in the codon, okay, instead of A in this codon, now it was replaced with G. Okay, it was supposed to be GAA, the sequence was supposed to be GAA on the mRNA. Okay, Now what happened? Instead of this A, who is coming and sitting? CAG was there. Means there is some mutation. One change was there. See, this GAA codon, it is going to code for GLU, glutamic acid, for example, glutamic acid, it is going to code for the glutamic acid, amino acid. Okay. Now, even this GAG, even this GAG also code for the glutamic acid. So, at the end product, it's the same. It's the same. Though mutation have happened, but the coding amino acids are going to be same. See, one amino acid is coded by, you have to understand, one amino acid is coded by multiple codons. There are multiple codons. Actually, there are 4 different types of codons which can code for one amino acid. So, GAA is going to code for glutamic acid as well as GAG is going to code for the glutamic acid. Both of them are going to code for the glutamic acid. So, in the protein, there is no problem. There is no problem. Absolutely, the mutation is silent, which is something good. No problem. But look, sir, GAA, it codes for glutamic acid. Good. But what happened? Because of the point mutation, instead of this, in the place of G, who is coming and sitting? Who is coming and sitting? U is coming and sitting. Uracil is coming and sitting. Now, this UAA, it is going to stop the process of transcription. Sorry, translation. It's going to stop the process of translation. It stops the translation. It stops the protein synthesis. Break. Stop. We will say, nonsense. Stop it. Right? Something like that, right? So, what exactly is nonsense? Nonsense means it's a stop. So, mutation happened, point mutation happened. Because of this point mutation, one nucleotide is replaced with other nucleotide. That nucleotide is giving signal to stop the protein synthesis, Nonsense, stop it. Nonsense, stop it. So, protein synthesis stops. So, there is an effect. So, whenever you have such kind of nucleotides, like such kind of codons, UAA, UAG, UGA, these 3 are the stop codons, sir. Because of some mutation, whenever you have this codon, these 3 codons are the stop codons. They will actually stop the process of protein synthesis whenever required. But because of mutation, when you have them, protein synthesis is going to stop abnormally. Such a problem. Okay. So, this is the second type. First type is silent mutation. Second type is nonsense mutation. Nonsense mutation is a type of point mutation. Next, third type is missense mutation. Missense. I have said you something, but you misunderstood. Okay. So, it was supposed to be GAA. GAA codes for glutamic acid. But instead of this A place, in the place of A, if C is replaced, if it is replaced with C, instead of A, it was supposed to be A over there. Okay, in the mRNA, there are codons. Instead of A, because some point mutation in the DNA, there is some mutation in the DNA, one thing is replaced, automatically in the mRNA also it's replaced, right? So, it was supposed to be GAA. Now, it is GAC. A is replaced with C. Now, what happens? GAA codes for glutamic acid. Whenever there is GAC, it codes for aspartate. Means in the place of glutamic acid in the protein, it was supposed to be glutamic acid. Now, who is there? Aspartate is there. So, one amino acid is totally replaced. Okay, now there is a misunderstanding. It was supposed to be GAA, but it is GAC. So, ribosomes misunderstood, tRNA misunderstood it, and now totally amino acid is replaced. So, that is called as a misunderstanding, missense mutation, amino acid changed. So, this is what classically happens in a patient who is having sickle cell anemia. Let me write here. Missense mutation is usually seen with sickle cell anemia. Sickle cell anemia patients are going to have the missense mutation. So, what exactly is happening in the sickle cell anemia, sir, if you ask me? Sir, in the DNA it was supposed to be, look, first let's start with the DNA. Okay, this is the DNA, sir. In the DNA, it was supposed to be like something like this, CTC. See, in the DNA, it was supposed to be CTC. The nucleotides are supposed to be CTC. See, whenever there is CTC here, now during the transcription process, in the mRNA, what do you have? GAG. Wherever there is C, there will be G. And wherever there is T, there should be A. That's, you know it, right? Okay. So, sir, in the DNA, it was supposed to be CTC. Whenever there is a transcription happening, in the process of transcription on the mRNA, during transcription, mRNA is going to form. So, in the mRNA, it should be GAG. So, this GAG codes for what? Glutamic acid. And this is something normal. So, glutamic acid was supposed to be placed here in the hemoglobin. In this protein, glutamic acid was supposed to be there. But there is a missense mutation. Now, missense mutation. What is that mutation? Instead of T in the middle, it was replaced with A. Whenever there is A over here, in the mRNA, U is coming and sitting. It was supposed to be A here. It was supposed to be A, right? Now, instead of that, U is coming and sitting. So, totally misunderstanding. Now, whenever there is GUG, this codon, this GUG codon is going to code for the valine. It was supposed to be glutamic acid over here. Now, instead of glutamic acid, now there is, what do we have here? Valine. Okay, valine. So, a hydrophilic molecule, hydrophilic glutamic acid is replaced by valine, hydrophobic thing. So, this is what is the problem in the sickle cell anemia. It's a quality of the hemoglobin is affected. So, instead of one amino acid, there is a different type of amino acid. This is the normal hemoglobin. This is sickle cell hemoglobin. This is the sickle cell hemoglobin because the glutamic acid was replaced with valine. Okay. So, here I have discussed about the three types of mutation. First mutation, that's a point mutation which is having three types. One is silent mutation, other is a nonsense mutation where there is a stop codon and the protein synthesis is going to be halt. And the third type of point mutation is called as a missense mutation. The example is sickle cell anemia. The glutamic acid was replaced with valine. So, total protein is going to be affected. Okay. So, now after this, the next type of mutation, point mutation completed. The next type of mutation that I am going to discuss here is frame shift mutation. Okay, frame shift. So, what exactly is this frame shift? There is a frame, there is something called as a reading frame. Okay, there is something called as a reading frame. For example, let me put it this way. See, this is mRNA. Here is mRNA. This is the mRNA, sir. Now, in this mRNA, for example, let me put it something like this. These are all the codons. Okay, these are all the codons. Nucleotides, I should say, these are the nucleotides. The three nucleotides together called as what? The three nucleotides together called as one codon. They represents one codon. Codon is three nucleotides together. This is the mRNA. Okay, for example, A. Okay, A U G U. Okay, AUG G A. Okay, next. Okay, C A G U A G, something like that. These are the codons which are normally present. Okay, something like this. So, these three are one frame. Okay, tRNA will come. tRNA will understand, AUG codes for some amino acid and that amino acid is going to be there. Now, these three GAA, they will code for some other amino acid. Okay, this CAG is going to code for some amino acid. So, these amino acids, whatever the tRNA is bringing, these amino acids are joined one another. Okay, by peptide bonds, the amino acids are joined one another in the ribosome. So, protein synthesis is happening. Okay, now because of addition or deletion of a nucleotide, for example, because of one reason, small reason is one nucleotide is deleted. So, see here what exactly is happening is this G. Now, if this G is gone, from there it is gone, means deletion. See, there is a deletion of that one nucleotide. When this one nucleotide is deleted, do you know what happens? Okay, now look here. Now, let me take this. See, now what just happened? This is gone. When this is gone, now A, U is gone. Now, this will become the codon now. AUG, 3 nucleotide, now this will become the codon. Now, AAC will become the codon. Means the reading frame is shifted. Now, the reading frame is shifted. Now, it's something like this. Now, there is no such thing as this one. Just let me remove this. Now, like this. Now, this AUG become the codon. It was supposed to be different. It was supposed to be AUG previously. It was supposed to be AUG. Okay. Here you need to understand one more thing. See, AUG, here also AUG because, okay, that's okay. Same amino acid, it will code for same amino acid, but see the next frame, it was supposed to be GAA, now it became AAC. Now, AGU, now it was supposed to be CAG. See, the entire reading frame is shifted because of deletion of one amino acid. It is totally shifted. So, entire reading frame is shifted, so the entire protein is going to be affected. Entire protein is going to be affected. So, that's called as a frame shift mutation, which is because of deletion of one amino acid changes the reading frame, changes the frame because the codons are taken as three nucleotides are considered as one codon. If you remove one nucleotide, that reading frame shifts. For example, let me add this thing. See, copy, let me copy this. Let me paste it here. Look here. This is something normal. Okay, this is something AUG, GAA, CAG, UAG, something like this. This is something normal. Okay. Now, what I have done, I have added a new nucleotide. Means there is addition. Deletion is completed. Now, what I am doing is, I am adding addition or insertion. So, here, look, I am adding one more nucleotide, A. Okay, I have added A, sir. One nucleotide is added. Now, what happens to the reading frame? See, reading frame is always 3. So, this 3 nucleotides are okay. Now, this AGA becomes one nucleotide. Now, this ACA will becomes the nucleotide. It was supposed to be what? It was supposed to be AUG. Now, here, yes, it is AUG. But because from next C, GA, it was supposed to be GAA. Now, it becomes AGA. It was supposed to be CAG. Now, it becomes ACA. So, the entire reading frame is shifting. As there is entire shift in the reading frame, the entire amino acid sequence is going to be altered. As amino acid sequence is altered, this is no longer going to be a normal protein. This is absolutely nonsense protein. It's absolutely totally wrong protein. So, the point which I want you to know here is, see, there are 2 types of mutations which I want you to know from this video. There is something called as a point mutation, which is of 3 types. Either it can be a silent mutation, either it can be nonsense or missense. Means one nucleotide is replaced with other nucleotide. Simple point mutation. One nucleotide is replaced by other nucleotide, which can either lead to silent mutation, nonsense mutation or missense mutation. But what is happening in the frame shift mutation? It's not replacement. It's not replacement. Either addition of a new nucleotide or deletion of new nucleotide. Addition or deletion is going to cause the frame shift. If there is shift in the reading frame, amino acid sequence is going to be changed. That will lead to total abnormal protein. So, these are called as the frame shift mutations. Okay. So, with this, the introductory video is completed. Here I have explained you all the basic things, basic terminology. What is a locus, exon, introns, allele, what is an allele, dominant allele, recessive allele, homozygous state, heterozygous state, codominance. Okay, genotype, what is genotype, what is phenotype, what are the types of mutations. These basic things we have discussed in this video. From the next video, we will discuss about the genetic disorders like Mendelian disorders like autosomal dominant recessive, X-linked dominant recessive, Y-linked disorders. Okay, so those things will be discussed in the next video. Hope the video is helpful. Thank you.